This is the first of 5 short videos that will provide an introduction to the immune system in preparation for the Intro to Immunology TBL. This first short video will address the differences in the innate and adaptive immune responses. We are exposed to a large variety of microbes all the time, and this includes bacteria, viruses, fungi, and parasites. And most of these microbes are harmless. They're commensal organisms that interact with us but don't cause harm to us, or they're environmental organisms that don't really want much to do with people. But a subset of these microbes are pathogens, and these are organisms that cause disease in their hosts. And so a key function of the immune response is to be able to distinguish self and non-self. So for example, we'd like to protect ourselves from infections and tumors, but not cause autoreactive responses against our own tissues. We'd also like our immune system to be able to distinguish between what is dangerous and benign. We want to make a response to pathogens, but we'd prefer not to respond, for example, to commensal organisms or food antigens or other things like that. So as we get into these questions of how the immune system distinguishes between self and non-self, and how the immune system determines what is dangerous and what is not, I'd like to start off with some key definitions. These come from probably your undergraduate biology courses, so they may seem a little familiar, but commensal organisms are organisms that benefit due to proximity to a host, but the host is unaffected. So one-sided symbiosis. A pathogen, on the other hand, is an organism that causes disease in its host. Virulence are the characteristics of an organism that allow it to cause disease, and an antigen is a substance or a component of that organism that is capable of inducing an immune response. It literally comes from the combination of the words antibody generator. Now, all of our leukocytes, or white blood cells, originate in the bone marrow from hematopoietic stem cells, and hematopoietic stem cells give rise to both common lymphoid and common myeloid progenitors. Common lymphoid progenitors in turn give rise to lymphocytes, including T cells, B cells, and natural killer cells, as well as a subset of dendritic cells. Common myeloid progenitors, on the other hand, give rise to myeloid cells, including neutrophils, eosinophils, basophils, mast cells, and monocytes, as well as a second subset of dendritic cells. The neutrophils, eosinophils, basophils, and mast cells are collectively referred to as granulocytes, and the neutrophils, eosinophils, and basophils are often referred to as polymorphonuclear cells because of the strange shape of their nuclei. Lastly, monocytes are present in circulation, but as they migrate into tissues, they differentiate into macrophages. Macrophages are not found in circulation, but are a key component of the innate immune response that is tissue resident. There are 2 main arms of the immune response, the innate and adaptive immune response. I'm going to go through some of the key differences between the innate and the adaptive immune response. First is that the innate immune response is really important for barrier protection. This really isn't a component of the adaptive immune response. The innate immune response is immediate, within minutes, whereas the adaptive immune response can take several days to get going. The cells of the innate immune response include all of the myeloid cells, as well as dendritic cells and natural killer cells, whereas the cells of the adaptive immune response are the lymphocytes, the B cells and the T cells, as well as dendritic cells, which are important for the induction of the adaptive response. There are key chemical components of the innate response that respond immediately, such as complement, which we will focus on primarily in these mini lectures. And the chemical component of the adaptive immune response is circulating antibodies, or immunoglobulins. The innate immune response receptors are encoded in the germline, and the adaptive immune response receptors are highly variable because they are generated through germline DNA recombination. This means that within the innate response, these receptors will recognize broadly expressed molecular patterns that are fairly nonspecific, whereas within the adaptive immune response, antigen receptors are very specific to unique pathogens. And lastly, while the innate immune response does not induce long-term immunological memory, the adaptive immune response does. So let's imagine an epithelial barrier that has been compromised by a wound or a scratch, and bacteria has been introduced into the tissue. These bacteria can be recognized in a variety of ways. One of those ways is through the activation of complement. Complement proteins can directly recognize bacteria and become activated to induce inflammation and pathogen destruction. On the other hand, innate immune cells such as macrophages and neutrophils can also recognize these bacteria directly, and they can be induced to make inflammatory cytokines and chemokines, and they can develop innate effector functions that allow them to destroy the bacteria, and this includes phagocytosis of the bacteria, degranulation, and killing of infected cells. So what is complement? Complement is a group of more than 30 proteins. They're found constitutively in plasma and interstitial fluid, and they're proteases but are inactive. They can become activated when they are exposed to a pathogen surface directly, or if they are exposed to a pathogen surface that is bound by antibodies. And when complement becomes activated, this leads to a proteolytic cascade that induces destruction of pathogens, innate cell function, and inflammation. Cytokines and chemokines, on the other hand, are typically produced by cells of the innate immune system and are induced upon recognition of pathogens. Cytokines are small secreted proteins that control growth, function, and or activation of immune cells, expressing the appropriate cytokine receptor. They can be constitutive or inducible. They are present in the innate immune response and the adaptive immune response, and they mediate many biological functions, including communication between cells of the immune system. Chemokines are a subset of cytokines that induce chemotaxis, or in other words, the ability of a cell to move from an area of low chemokine concentration to an area of high chemokine concentration. Chemokines essentially orchestrate the inflammatory response by inducing cells to migrate to the site of injury or infection. There are some key effector functions mediated by cells in the innate immune system. The first of those is phagocytosis, which is mediated by macrophages, neutrophils, and eosinophils, in which these cells take up foreign particles, including whole pathogens, into endosomal vesicles called the phagolysosome. And they're just within the phagolysosome, they're able to destroy those pathogens. Another key function of the innate immune system is degranulation, in which cells of the innate immune system release inflammatory granules that mediate inflammation and cytotoxicity. Some cells in the innate immune system can release their DNA into the extracellular environment as they die, creating what are called DNA traps. The DNA is sticky for pathogens and full of antimicrobial molecules. And lastly, some cells in the innate immune system, particularly natural killer cells, can have direct cytotoxicity, whereby they can recognize virally infected cells and by means of release of cytotoxic granules, kill them. These innate cells tend to have enhanced specificity for certain types of pathogens. So for example, neutrophils and macrophages are important in the antibacterial and antifungal response, as well as the response to single-cell parasites. Neutrophils are the most abundant leukocyte and comprise 40 to 70% of leukocytes circulating. Macrophages, of course, do not circulate, as I mentioned previously, and are found only in the tissues. Natural killer cells are largely important in the antiviral response, whereas eosinophils, basophils, and mast cells are important in the anti-helminth response, or against worm parasites, as well as in the induction of the allergic response. Now let's go back to our injured skin, in which we have the entry of bacteria into the tissue. Now, of course, recognition of this bacteria by a macrophage will allow this macrophage to start producing inflammatory cytokines. However, dendritic cells are also present in the tissue, and when they recognize those bacteria, they can migrate to lymph nodes where they will activate T cells. Antigens drain from these tissues into the draining lymph node. So here's an activated dendritic cell. It has been busy eating up viruses or bacteria, whatever is in its environment, but it is now ready to migrate via a lymphatic vessel into the draining lymph node. Bacteria, free virus, free proteins can also travel via the lymphatic vessel into the draining lymph node. Once there, these dendritic cells can induce the activation of T cells. There's two key and broad subsets of T cells that we'll be talking about throughout our study of immunology, and that is the CD4 positive T cell and the CD8 positive T cell. When CD4 positive T cells are activated, they differentiate into what are called helper T cells. These helper T cells secrete cytokines that activate innate cells and B cells, and they have really important antibacterial, viral, fungal, and parasitic functions. CD8 positive T cells, on the other hand, differentiate into cytotoxic T lymphocytes. And while they also can secrete cytokines, they play a really important role in killing virally infected cells and are a key component of the antiviral response. B cells, on the other hand, can be activated by exposure to free antigens. These can be soluble proteins or surface antigens on viruses or bacteria or other pathogens. When B cells become activated, they can differentiate into plasma cells that produce secreted antibodies. And these antibodies can neutralize foreign pathogens to prevent their ability to infect. They can activate innate immune cells, and they can activate complement. Importantly, the adaptive immune response forms long-term immunological memory. A fraction of activated T and B cells can persist for many years as memory B cells, and if they ever see the antigen again, they can generate a robust secondary response that is increased in size and quality compared to the first exposure. And this picture on the right illustrates that by showing 3 different immunizations of first, second, and third boost, and showing the impact of each of these immunizations on both the size and the affinity of the antibody response. That is the end of this Intro to Immunology lecture. In our next mini lecture, we will delve into some of the key effector mechanisms of the innate immune response.